So, ladies and gentlemen, it's been suggested to me that we actually just start the session in the hope that the fact we've started might bring people into the room rather than waiting for the other way around. So, first of all, hi. I'm Jeff Hall. I'm a medical oncologist in Leeds. Leeds were one of the centres that got Eden funding to do an OMOP mapping of its data, and that early support has led to Leeds being part of local, national, and now international studies with its data. So I would encourage any organisation in the room to follow that same sort of process. So I'm delighted to be one of the people asked to chair this session, and I'll just pass the mic to my colleague Ofran.
Thank you very much, Jeff. I'm Ofran. I'm a clinical academic at Great Ormond Street Hospital, and I'm also a postdoc researcher at UCL Institute of Child Health, and most of my work is involving routine collected data. So I'll introduce the first speaker. Jenny is a clinical academic and a lecturer at Queen Mary's University Hospital. She's presenting today on behalf of Hiba, who couldn't be here. Hiba is one of the data scientists at Barts Life Sciences, and she uses part of her work in real-world data to benefit patients. Over to you, Jenny.
Perfect. Good morning. So yeah, as has been said, I'm presenting today on behalf of Hiba Junaid, who's one of our team in Barts Health, who has been pioneering this work, and also on behalf of all the others in the Heron network. So I do feel a little bit of pressure to make sure I represent everyone who's been working in this network over the past year to talk about this study. So when thinking about the studies that were undertaken in Heron in year one, the real focus was on a national stage and things that were interesting across the national board. So this question was looking into high-cost medicines, and we know that there's an increase in spending on medicines by the NHS in general, with a almost 44% increase over that time spent. And we know that the majority of these high-cost medicines are prescribed as inpatients. And this is of interest because traditionally less has been known about the spend on medicines that are prescribed during hospital stays in comparison to that in primary care. And the other question that came out was whether there was a potential disparity in access to medicines, and also that there are individual funding requests that have to happen for some medicines, and whether we could see if there was a difference across our network for that. So this was a study that was undertaken within the Heron network using the coordinating centre in Oxford and along with our partners across these centres. So the plan was to go in our collaborating centres, and we looked at data from the 1st of January 2022 to the end of data that was available. And we looked at the January 2025 list of NHS England high-cost drugs commissioning lists, of which we were able to look at 249 of those drugs together and looked at them regarding their ATC classification groups. And then we made cohorts of new users of these drugs, looking at their age, their sex, and conditions of interest that came into their data 14 days before or after the use of the drug. So I'm sure you're super excited to see this quite busy table. So what this is these are the top 10 drugs in each of the 4 centres that we had. And if I highlight the top 4 for you across the groups, I can tell you a bit more about them. So Barts, that's my centre. So we're based in East London. Our top drug was this drug that is used predominantly in renal replacement therapy to increase red blood cell count for anemia. Great Ormond Street, of course, a leading centre for childhood paediatric care. Their greatest use was mycophenolate, which is an immunosuppressant drug used for solid organ transplant. Lancashire, up in the north, this one I'm going to get wrong, west, they also were using the drug for renal replacement therapy. And UCLH, their largest new user drug was tadalafil, which is used for pulmonary hypertension as well as erectile dysfunction. So from that perspective, it's more from the pulmonary side that they were leading. The other leading drugs were tacrolimus, again an immunosuppressant, and infliximab, used for a variety of rheumatological and colorectal diseases. The other thing that we're able to do across the board for different drug classifications and groups, we will be able to make plots like this. Now, I appreciate this is super busy, but the bit on the left shows you the number of new users, then we have the median age, and then the proportion of patients that were male. And you'll see that there's some dots that stick out a little bit. Let's see if my laser can work. It can't. So on the very left of the middle column, which is the median age, you'll see that there are some that stick out with a very young median age. And one of those would be Sidovudine, which is used for to prevent HIV transmission from mum to baby, as well as Pilivisudumab. I've tried to say that, at the bottom, and that's used for RSV infection in children who have very severe immunocompromised states. We're also able to do this looking at anti-cancer agents, and this is where you can see the proportion of agents we are able to look at, and also looking at the amount that were prescribed between the hospitals. And you can see here that this really shows how much cancer work that UCLH is doing, in addition to in the previous slide, looking at anti-infective. UCH has a large proportion of HIV and AIDS patients that they are looking after. In terms of conditions, we were then able to drill down a little bit more and look at the different groups. And here what we're able to look at is the drugs that were used for different types of cancers. So you can see here that we have, obviously, there are some here that are predominantly male or female cancers. So looking at that proportion of sex and age is less useful here. But we're able to look at the top drugs that are prescribed across different areas of the country and able to pick that out. We can see here that we can see the difference between the patient profiles that we have in the different hospitals based on also the use of immunosuppressants. So here, a variety of patients who have received organ transplants, in addition to those who have immunological diseases like ulcerative colitis and Crohn's disease. And this is where you can pick out the use of infliximab and mycophenolate that was picked out in that top 10. So overall, what we found is that the largest groups of drugs that were used that were from the high-cost list were antifungals, immunosuppressants, monoclonal antibodies, and antivirals. But what it really showed as well is that we were able, within the HERON network, to use interoperable data to enable real-world surveillance of drugs. And this is something that we hope will be of value for regulatory and health technology assessments in the future, as well as a foundation for looking at outcome surveillance in future. Of course, it's all the people we have to thank are from the different centres who've really been able to run the study, develop it, and then give us this information today. Thank you.
Brilliant. I hope there might be a roving mic on the floor. Is there a roving mic on the floor? No, maybe we're using this one. Oh, there's Ed. Have you got a mic with you? Are there any questions from the floor for Jenny? Yes, please.
I don't think it deserves some work. This is a very nice characterization. Do you have plans to use this information about the treatment of the patients to also derive some hypotheses and test them? Or how do you think this project will go on?
So, correct me if I'm wrong, but I believe the reason initially for doing this was actually about understanding from a national level for the NHS about which things are actually used in secondary care, because we have great use with CPRD, but that had initially been an area where that there wasn't as much information. So this was both a use case and to provide that information back to the MHRA and to the NHS in general to identify those areas and to look at disparity. It wasn't necessarily to then go into a next question, but of course, now we have those phenotypes, we have those groups, we very much could do. Yeah.
Anybody else with a question? If not, Jenny, can I ask, it struck me that there was quite different use of these different drugs with different hospitals. Do you think that is just about different patient populations, or do you think there's anything different in policy of use of those drugs? Did that bring any of that out? Because, I mean, particularly the darbepoetin or some of the anti-cancer drugs, I think I'd have expected to be a bit more uniform.
I think that's really interesting. And I think for that information at the moment, we haven't compared that to local policy guidance. And it may be one of those areas where we can reach out to local clinicians who are subject matter experts in those centres to say, is there something that is limiting you for commissioning, or is it because of local clinicians feeling that they wish to use that more, or is it something specific about that patient population? Obviously, at the moment, we can make assumptions, but we can't necessarily drill down any further as to why that's a difference at the moment.
And for any centre out there who's interested in sort of running this code against their data, is that code in a public repository that?
Oh, yes. Yeah, so everything's out there, and it's very much, at the moment we're looking for people to join us. It's not something that is expanding for now, but all of that information would be out there and available to assess and look at the code.
Fantastic.
Site to site, the genomic testing, which is really important in cancer indications, tends to be different site to site a little bit. Have you got that data to check if that might be underlying some of the differences in the cancer medications?
That's a really good point. We haven't included genomic or genetic data in this characterization. Again, I think it probably feeds back into what Jeff was saying about speaking to local subject matter experts. We know what each centre, their patient profiles are like overall, so we can tell the general populations they serve, but we haven't drilled down into that at that stage. So it's a good point.
Brilliant, Jenny. In the interest of time, I'm going to draw that to a close, but thank you very much. And if anybody wants to take Jenny on, then go and find her later.
Right, our next speaker, are you a pair or just you? No. So the next speaker is Amani El-Belushi, together with Yuki Wang, who are PhD students at the University of Manchester. They're collaborating with Lancashire Hospitals, is that right? And they're looking at research focused on antimicrobial stewardship in real-world evidence.
Yep.
Over to you.
Thank you. So can I, just move like this? How to move the slides? Okay, thank you. Thank you so much. So good morning, everyone. It's my pleasure to have me here, and thank you so much. Actually, my name is Amani El-Belushi. I am a PhD student with my fellow Yuki Wang. We both PhD students at the University of Manchester. We're having our project with collaboration with the Lancashire Teaching Hospital. Our project is about the antimicrobial stewardships. So actually, Lancashire Teaching Hospital, definitely they are leveraging millions of prescribing data points every day. So after transforming those data into the OMOP common data model, we've been able to have some actionable insights to combat the AMR. So what is AMR? So AMR is when the microbes stop responding to the treatment. So, and this is actually derived usually by the overuse of medications or misuse or poor infection control. So switching from IV to oral switch is one of the stewardship activities that's recommended to manage the AMR. So, and this is because IV therapy usually it is preserved as the last resort option. So prolonged IV therapy usually increase the risk of complications, increase the cost, and definitely prolonged therapies and prolonged stay in the hospitals, this will affect the resistance. So our study, we conduct a retrospective cohort study at the Lancashire Teaching Hospital. The data analytics team at the hospital, they help us in providing us with a secure environment, analytical environment, which enable us to have a dedicated platform to run our analysis and answer our research questions. So our population, including the inpatients, for 5 years, from 2020 to 2024, who've been given at least one antibiotic, IV or oral. So we came up with 860 patients who've been given millions of antibiotics, IV and oral, and around 60% of them being given intravenously. So to illustrate our key findings, we started with the prescribing patterns. So as you see here, the coamoxiclav is dominating with the high prescribed antibiotics in both routes, IV and oral. And also there is some broad spectrum antibiotics being also dominating, like piperacillin, diazepam. So on the other hand, there are some oral options being underused. So we indicate here there is a high reliance on broad spectrum IV, where there is some opportunities to de-escalate. Again, here we took the top 5 antibiotics per infection. So again, you can see in the deep blue, coamoxiclav is the most prescribed antibiotics across all infections. So, and although the prescribing is condition specific, but still there is high reliance on the broad spectrum, especially in the respiratory infections. So that's why we took the coamoxiclav for further investigations and further analysis. So we created those flags. So if you can see here, we have only 18.9% switched to oral within 4 days as the guidelines recommended, as it's shown in the blue bar. The yellow bar is indicating the prolonged IV use, which increasing throughout the 5 years. And also we have some also delayed in switching as shown in the green bar. So here we have the daily trend analysis for the IV switching for the antibiotic medications following starting with the IV therapy. So most of patients started IV, but there is a slow switch. Actually, around 20% only switched after the day 4, which is the recommended guidelines. So as it is shown in the blue color. And the stop is happening only after first week of prescribing. So this is indicating a very slow switching from IV to oral. So to conclude, actually, there is a high reliance on IV broad spectrum antibiotics, and the switching is very slow in most of the cases. So this brought us with a good opportunities to start a new stewardship with in the hospital to give a chance to better care and better control to the AMR. Actually, we're working very closely to the microbiologists and the clinicians at the Lancashire Teaching Hospital to design some interactive dashboards that will help the healthcare providers and the microbiologists with a very nearer time knowledge that will help them to optimize antibiotics prescribing. And also we are developing a prediction model that will help them to decide when it is safe to switch from IV to oral. Thank you so much, and that's it.
Mani, thank you very much. Once again, are there questions from the floor?
Yes, hello. I'm just curious, what reasons would there be to, for example, prolong the use of IV-delivered antibiotics? Are there good reasons to say, no, we're not ready to switch to oral yet? I'll keep them on the drip.
Yeah, actually, I don't know if I understand your question, but if it's why we need to switch to oral instead of IV?
I know that eventually there's a preference to switch from
Yeah.
IV, but are there good reasons? Are there reasons?
Yes, we need to check the stability of the patients first. I mean, not everyone it's not like a standard that after 4 days, patients need to be switched. There is some assessment need to be done for the patients such as they need to check if the patients they can swallow, if they are nil by mouth, or they have also some clinical. I think Yuki can answer this question because she's doing this.
Sorry to interrupt, because switching is my topic, so I can answer some aspect. IV to oral have some benefit if patients with frailty stay longer in hospital, they can have some IV device-related infections, and also can be hospital-acquired infections. Also from the economic reasons, IV-related device will cost much more than oral tablets. And also IV to oral switch is a policy in UK for better antibiotics resistance topic.
So it should be quite a quick question. So suppose I was looking at the dates that you had, like, were you able to control for people with COVID-19? I suppose is the question, and that might be some of the feedback you get from the clinicians at Lancashire.
Yeah, actually, we didn't exclude COVID-19 until now, but this is one of the steps that we're thinking about, because we cannot predict like how relevant was the prescription at that time, but it is a very huge data. We found with some antibiotics there is a drop during 2020, 2021, but some of the drugs we have also very high. So it is not very relevant to the right prescriptions. So this is one of the things that we're thinking of exclude patients during COVID-19 or who have been diagnosed with COVID-19.
And perhaps as a final question, do you have any perception of how closely your data follows microbiology policy in your hospital? Because I have to say, I look at that and think, wow, that's completely different to what my hospital's infection management pathways would be. So is that because your clinicians are ignoring your guidelines, or because your guidelines are different to our guidelines? Do you have any view on that?
See, actually, this is the data that we've got, and this is what we're doing. Our next step is to check with the microbiologists. We are to have a meeting very closely to them, very soon with them, for them to confirm that what we have found, it's very similar about what they are doing.
Brilliant. Thank you very much. I look forward to seeing the next stage.
Thank you.
Thank you very much. Next speaker is Megali Rufia, who's the data lead at the Health Innovation East. She oversees the data lifecycle for the East of England secure data environment. She has over 15 years of experience in developing and managing data infrastructures, and her expertise spans genomics and more recently health data.
Thank you. So good morning. So I am coming from an NHS secure data environment kind of topic. So in particular, the East of England SDE. So I'll give a quick overview as to what the SDEs even are and what the East of England SDE is doing. Then a kind of focus on a heart failure study we did and why OMOP was really helpful there. And then why it's only the first step and what we're working on next. There you go. I've even forgotten what I put in my slides. So well done. So the East of England SDE is part of the Data for R&D program, a network of England-based SDEs. We're focused on the East of England region. You had the talk about HDRS this morning. Rumors are that we'll be kind of subsumed in that. So the SDEs remain just under a different governance. We are in the East of England. We are hosted by Cambridge University Hospitals as our lead organization. So the idea was to move from a data sharing to a data access model. So SDEs are to become the prime way of accessing routinely collected NHS data for research purposes. In the East of England, we do not have an existing data lake. We did not have anything to build on top of. Instead, what we focused on was a technical solution, so a cloud-based infrastructure in AWS built around the 5 SAMES framework, compliant with SATRA specifications, which are the gold standard for TREs, and is ISO 27001 certified. So that's the technical platform. It means we control what goes in, what goes out, who has access to what, for what project. And the data remains with the data controllers. We extract the relevant subset for a specific project, give access to the researcher to that subset only. And so to kind of demonstrate the capabilities of the platform, we focused on an initial use case around heart failure, and in particular, the hospital readmissions for heart failure. So various papers claim there's a 20% readmission rate within 30 days. So patients come back a little bit quicker than we'd like. And so the idea was to, first of all, confirm this on English NHS data. Is it really 20%? What does it look like? Are there common key factors? And kind of further down the line, looking at, can we develop a risk prediction model based in the hospitals that could help us spot those patients and provide an alternative care pathway. So we looked at all the hospital admission data, so all the physical measurements that were done, the test reports, the demographics, lengths of stay, what procedures they were going through, all the diagnostic codes, so primary reasons, but also any additional history. And we did that across 3 hospitals. And that's where our challenges started. The data was provided by the hospitals de-identified at source, and we pulled it into the environment. These were 2 different EPR systems and 1 hospital without an EPR system. So we mapped all the data to OMOP to facilitate this study and be able to compare apples with apples, essentially. We're having some really interesting initial findings that the readmission rate, for example, is between 10 and 15%. Which means it's lower than what the paper said, but also very different from one hospital to another. We have one hospital on the East Coast, so coastal area, very rural. We have Cambridge in the middle, very different population. And then we have one hospital more towards the centre, or the west part of East England, which is the centre of England. So very different populations, very different readmission rates, and we're also noticing different demographics. So that's the kind of next step we want to do with this data. But what we're missing with this data is because it was de-identified at source, we did not have the linkage. So we do not know, is the difference in readmission because this is what the local population was, or do some patients go to different hospitals in different circumstances? Is one hospital specialized in heart failure treatment, so all those elective appointments would be more frequent in one hospital compared to the others. And that takes me to the last bit, which is what we're working on at the moment. We want to standardize this OMOP mapping, and again, to prevent us, well, to avoid having to build a data lake. We're still keeping the data within the data providers, but we're supporting them, and we're having help from the Hive to map their data to OMOP locally within their GDPR boundaries, so they have that local asset that they can query. We're also working with Fitfile to kind of link the patients between the data providers in an encrypted fashion, which I do not understand, but leave it to the experts. This means that we can have one data set, but separated across 3 nodes or more when we get more data providers on board. And it means that we can support cohort discovery queries through the OHDSI UK gateway. And rather than saying we have 100 patients in one hospital, 200 in another, and 150 in the last, we can actually surface a cohort of 500, which is way more powerful. And it will also be the way we'll extract the data. So we'll be able to provide the data mapped to OMOP alongside the original data. So we can essentially use OMOP for all that cross data sets analysis, kind of get the wider trends. And if there are any doubts, the original data is still there for more in-depth exploration. And that was the last slide. So thank you all for listening. And I'll take questions.
Magali, thank you. So once again, questions from the floor?
Hi. Thank you for all your hard work. It looks like you're taking in a lot of sources. I'm over at Oxford. My name is Kristen Koska. So I have a question for you. You're using a third party, it sounds like, to help you with mapping. Are you keeping track of just the learnings from that mapping process? You have some very interesting feeds that might face some concept prevalence issues or, concept mapping issues. Or what are you doing with that insight? And I say this as a long time listener. I've been doing the Odyssey for 10 years, so you might have some knowledge for your peers.
Yeah, so it's still early stages. We have now synthetic data mapped for 3 of those sites. So we're starting to look at the kind of comparing what were the insights. The next step would be to bring our data providers together. So kind of, as we've discussed in some of the previous talks, is kind of go back to the clinicians, to the subject matter experts. What was the logic behind it? Is what we mapped actually an accurate representation of what happened in the hospital, and then kind of look at across sites, well, why are there differences? Where do those differences come from? And can we together kind of push towards a little bit more harmonization at the source?
If I could ask a follow-up question to that point, where does the OMOP ETL happen? Does that happen in the trust, in the fit file layer, or in the SDE?
So we're basically, each trust has its little ring-fenced area, which is still within their GDPR boundaries, but is a copy of the of their local system, so that it doesn't impact direct care, and that's on that database that the OMOP mapping happens. So it's still within the trust.
And you have a very clear fit file dependency in that. Yes. So if Hospital 4 wanted to contribute but didn't have a fit file node, can they not contribute to that work?
So part of the partnership we're doing with the data providers is support them to get that fit file installation.
So you are creating a fit file dependency, and
yes.
Fine. Are there any other questions from the floor? Excellent, Magali. Thank you very much.
So our next speaker is Dibya Mohanty. Is that right? Dibya has a master's degree in genomic medicine from Trinity and currently doing her PhD at University of Limerick. Her doctoral research focuses on the tissue specificity of cancer, leveraging genomic data to looking at mechanisms that drive tumor progression.
Good afternoon. So this mouthful of a topic right here is quite an interesting work going on at the eHealth Hub for Cancer at University of Limerick. It's a collaborative work, and you can see there are different great mentors and amazing minds working at it. I'll be presenting you an overview of what we have been doing so far. So first, to start with, to advance cancer research in Ireland, eHealth Hub has been established. It's a cross-border collaboration established with a mission to unify and create an e-health strategy for characterizing blood cancers all across the Ireland. Now we are concentrating on 3 specific blood cancers right at the moment: AML, MM, and CLL, each with a dedicated lead institution and research focus. Currently, we have, these are our clinical sites, 11 to be precise, and these are our academic sites. For this presentation, we will be focusing on AML alone. Now, let me walk you through why AML is such a complex disease to decode. To start with, AML is a disease of clonal expansion. Immature myeloid blast cells take over the bone marrow and spill into the blood, blocking the normal hematopoiesis. This shows why the patients who show up are seen with anemia, bleeding, and infections. Now, this is very aggressive and a rapidly growing disease and requires immediate attention. Moving on to the etiology. Oh, I think it's been zoomed for some reason. It's been zoomed for some reason. So moving on to the etiology, we can see that the genetic mutations are very common, like we see FLT3, NPM1, IDH1, 2. These mutations very largely dictate the therapeutic choices we make. But cytogenetic abnormality also dictates and creates a crucial role in the etiology of the disease. But the real challenge lies in the genomic heterogeneity. Within patients, we see that the disease looks different, but between patients, the case is even more complex. And this is the basic reason why it is so hard to decode. Now, trying to understand this, it's a loop. So the disease we started and diagnosed and defined at diagnosis is no longer the same at its relapse. Having said this, we understand that the biology of this disease is very dynamic. So our therapeutic choices and transplant-making decisions have to be so too. Now, this is a statistic that shows that between 2004 to 2019, the incidence rate of AML has increased for 3 to 4 new cases per 1 lakh patients in Ireland. And it also shows a very close relation that the disease is mostly seen in adult patients, giving us a direct link between the age and the disease. Now, understanding this, we conducted a comprehensive landscape analysis across 5 key clinical sites in Ireland, and the results we obtained gave us gaps that needed to be mended. So first, we saw that the majority of data are still paper-based, which majorly hinders the clinical analytics we plan to run. We also saw that the hospitals use numerous different data, and thus the data are very heterogeneous in nature. Third, we saw that different vocabularies, different ontologies are being used. And last, that the data sets are isolated and disconnected. Based on these findings, our central goal is to harmonize and create a standardized data. This will not only lead to data harmonization, but also enable fair sharing for clinical evidence generation. To accomplish this, we are adopting the OMOP Common Data Model as the standardization framework for our All Ireland studies. So we are currently implementing several steps to standardize the data across all sites. First is to develop a data dictionary that tells us which data to collect, required formats, standards, tables, etc. Then to map the data fields from the data dictionary to the health information systems. Then we have when we have to set up a database that defines the data fields from the database with a structure that directly aligns with the data dictionary. Then to conduct a thorough gap analysis and data quality review using various combination of tools within the OHDSI standards and manual audits. And finally, with a collaborative sharing agreement in place, we are designing a data blueprint which should be used to build the ETL scripts needed to process and standardize the data. Now, to ensure transparency, both the data dictionary and the ETL scripts are maintained in a shared GitHub repository. This approach allows us to track different versions effectively and ensures our tools are reusable for future projects. Now, when talking of the data dictionary, particular attention was given to the genomics table. Genomic alterations in AML are very crucial and complex. As we understand that genomic alterations in AML are both complex and clinically essential as they inform targeted therapies and risk stratification. Since our data is collected manually, the table had to strike a balance. While capturing all of the relevant information, it still had to remain manageable for the data entry personnel. To ensure interoperability, the table was designed to be mappable to OMOP CDM concepts. In shaping its structure, we reviewed existing data dictionary from the international AML networks, including the Harmony Alliance and the Pediatric Cancer Data Model. We also understood the genomics representation in OMOP Common Data Model, and we consulted with hematologists actively treating AML patients, so our design is in alignment with the entire process that's going on. The resulting first version of the genomics table supports the recording of the gene variants, fusion events, and structural variants. For each category, we included a curated list of alterations most relevant in AML, while also allowing free text entry of additional variants to capture emerging or rare findings. This work has been done by a postdoc at eHealth Hub, Dr. Marina, who's also present here with us today. So while understanding this, we are also making significant progress in developing a robust ETL pipeline for data standardization. Our development followed 3 key stages. First, we used Cynthia to generate 2031 patients and adjusting its AML module to simulate a realistic cohort. However, we understood that the data we generated largely lacked the granularity we needed for our research. To address this, we adapted our ETL script using the Mimic4 dataset. Its rich granularity definitely helped us assess different model and AML variants such as relapsed cases and secondary AML. However, we also understood that both Cynthia and Mimic4 largely lacked genomic data. So to make our pipeline more comprehensive, we made a final adjustment using a dataset from a study focused on mimicking clinical trials with synthetic genomic data from generative AI. Although the genomic data we used for this data set and understanding was not highly complex, but incorporating this, we also found that it paved way for us to be able to handle the real-world complexities of the genomic data coming our way. Our immediate next step is to apply a refined ETL script to the retrospective patient data collected from 2020 to 2022 across the key clinical sites in Ireland. However, we also understand that this will involve minor refinements in the scripts. This work has been done by a postdoctoral student at eHealth Hub for Cancer, David Okpuru. Now, in summary, by integrating longitudinal data, we expect that this framework will support more precise treatment, and ultimately this project will deliver a federated, fair, aligned clinicogenomic framework for AML research in Ireland, as well as it will pave the way for international network studies in AML. Thank you.
Dibia, thank you very much for that. Once again, are there questions from the floor? If not, can I just say that's really impressive. And at one level, it feels like you've tried to do something that I think is quite hard, which is very admirable. But hard can come in two directions in OMOP. One is the number of centres and the number of different systems and the number of different approaches. The other hard thing to do is the number of data items. How deep is your data set planning to be?
Okay. First, thank you for the appreciation, but I'm not solely responsible to be taking it all. It's a collaborative work, and major work has been done by my colleagues. As I understand now, we are expecting to expand to all of the 11 clinical sites currently we are, like eHealth Hub is considering. And once this pipeline is robust enough, our major term goal is to deploy that pipeline across all of the 11 clinical sites and gather the real-time data and structure it and use it as planned.
So you've demonstrated patient demographics, so ages of the patient and gender, and then you've described tumour characteristics. Are you also looking for treatment pathways data as well?
Once we will be able to characterize the pipeline robust enough to be able, then we'll be able to work with the real-time data. Right now, we have no real-time data as of to work with.
Okay,
we have not started anything to do with that.
Are there other questions or comments from the floor?
Thanks very much. You put a lot of genomic data into OMOP. I know that there are sort of OMOP expansions for genomic data. Is that what you used, or did you make your own set of tables and you made your own?
Okay, so I'm sorry, but I might not be able to answer that. My colleague Marina here is the best one to be able to answer that question. Marina, please.
Hi. Yes, so we are using the mainly this OMOP genomic vocabulary that has been developed by the oncology working group. And I think that's mostly what is available in OMOP in general. So it only works if you are working with cancer, actually, I think. But for us, I think it will work well.
And any last questions from the floor? If not, let's move on. Thank you, Divya, to our final speaker.
Thank you. Our final speaker is Gianluca Fabiano, and he's a senior health researcher in health economics at NDOMS in Oxford. He works with the health economics outcomes research team. Is that right? I said. He focuses on informing decision-making through economic evaluations using real-world evidence.
Thank you. So, thank you, everyone. It's good to see you, to be here as a health economist in this room. This study is part of a bigger effort that our team in Oxford is undertaking to conduct analysis using OMOP in health economics. And last year, we conducted a distributed network analysis using data mapped to OMOP from 6 European countries. and we estimated primary and secondary care resource use. We demonstrated with that study that it's feasible to conduct this kind of analysis, but in this study specifically, we wanted to focus on how accurate these estimates are. So the objective was to compare resource use and costs, specifically in the UK, using CPRD data mapped to OMOP, and to compare and see how equivalent they are. We conducted a retrospective cohort study using data from 2010 to 2018 about women with the first fragility fracture in postmenopausal, and for up to 2 years follow-up. We were interested to know how many encounters in primary care these women had and the specialty of the provider. So who do they see? Do they see a GP, a nurse, or just an admin contact? And then to estimate costs. Now you know that in administrative data sets, cost usually is not within the data. So we use unit costs from outside, and we attach to all of these encounters based on the specialty the unit cost to estimate the overall economic burden. We conduct the same analysis in OMOP and in source CPRD, and then we compare using means and confidence interval from bootstrap samples. In terms of results, you see that we obtain a very similar, well, I think this is actually, I don't know if it was updated in the slides, but in the new slides that I updated a few days ago, you would also see the size of the cohort. So the size of the cohort between source CPRD and OMOP is very similar, it's almost the same. But as you see here, there is an exact match in terms of age, follow-up time in terms expressed in days, fracture site, and healthcare users. These are the percentage of people who see, who have a clinical encounter. So if they see a GP or a nurse at least once, they would be a user. If they only have an admin or have no visit at all, they would be non-users. So you see that there is the estimates that we derive are quite equivalent. Here instead we see to the left the mean encounter per patient per month. As expected, there is a peak at the beginning right after the fracture, and then downgrading over time. And OMOP and source CPRD produce the same estimates. To the right, instead, you see the mean cost per patient per month. And again, the trend is similar, but you see a slightly small gap between the estimates from source and OMOP, with source CPRD producing a slightly higher cost per month. And I'm going to tell you why in a second. So here we see a breakdown of the mean encounters by specialties. And the first thing you might notice is that in source CPRD, there are specialties that we don't see in OMOP, for example, the pink one. And also you see that in OMOP, the black bar represents specialties that are unassigned, uncategorized. So to the left, instead, in the dendrogram, which you could see also in the poster that will be later available, you see in the outer circle that these are all the specialties that are available at source level, in source CPRD. And their mapping into OMOP is in the inner circle. So you see that, first of all, there is a more granular definition of specialties in source CPRD compared to OMOP. And also 2/3 almost of them do not have a matching concept. That's what is in the black circle. So it means that for 2/3 of them, we don't know what kind of unit cost to assign, and in this case, we assigned a mean unit cost for all of them. This explains the small gap in the cost that you saw in the graph. So to conclude, estimating primary care resource and cost in OMOP produce largely comparable results. There are minor differences in the cohort composition, which is due to the quality standards that are applied in the mapping, and in the specialty classification, with some codes in source CPRD that lack OMOP equivalents. Refining this specialty vocabulary can improve the accuracy of resource use and cost estimation and support our role in bringing OMOP and health economics closer. Thank you very much.
Gianluca, thank you. So questions from the floor? Danny.
Thank you, Gianluca. So when we talk about different common data models that exist in Europe, one of the things that I always say about OMOP, which is one of the reasons I like it, is that it preserves the source concepts on top of adding in the standard ones.
Yeah.
Will you not be able to use that information in your analysis if you really need that level of granularity? Because the data is there, right?
Yeah, it is there. But as I said, then if you were to run analysis in a federated way, so in each country you would have different values for the different specialties, which somehow makes it harder to estimate and run a single code in all the settings. So if we just rely on the specialties that are standardized, we would face this sort of limitation in a way. But of course, in our case, we were able to estimate costs using source specialties as well. So it is possible, of course, to bring in some information. That's why we were able to apply a unit cost that is a mean, because precisely we could see what is the source that maps into the standardized values. Yeah.
Thank you.
Chris?
Yes. Thank you for that. It was really interesting. I'm just curious by your comment on there saying that there are fewer women in the OMOP cohort due to quality standards. Can you comment on that a bit more, please?
Sorry, I couldn't hear.
About saying that there are fewer women in the OMOP cohort due to mapping quality. Can you comment on that a bit more?
Yeah. So in OMOP, we identified something like 22,900 women, so the size of the cohort, compared to 23,100. So it's a minor difference. And that's due to the quality standards. So essentially, when the mapping is done, some records are dropped for some quality checks. So perhaps the death is occurring before, after the record of a fracture or something. Like, probably Antonella can also explain because she's in charge of the mapping here. But yeah, it's due to that, which, right?
But why is it affecting women more than men?
Well, this study is just about women, yeah.
Okay, maybe I'll ask you later.
Okay, so 12:30, almost bang on the dot. Gianluca, thank you very much.
Thank you.
I understand our next moment is lunch, which is that away. Thank you very much, everybody.